Sir, you're live now. You can start.
All right. Good morning, everybody. All the students who have joined us for the different courses of MOOC. Welcome to the live session, which is the first live session of this semester. I'm Sanjeeva Srivastava, professor at IIT Bombay. I'm instructor of your course on proteomics, gel-based proteomics, as well as the proteogenomics courses. Today, we thought to interact with you live. And during this live session, we will also give you some of the experimental setup what we have in our lab and how these kind of experiments can be performed in the laboratory, right? Of course, you have been listening our lectures, but today we thought to give you the feel of the gel-based proteomics and mass spectrometry-based proteomic analysis. So before we start, I thought it would be a good idea for you to know who are the TAs who are running the show behind, and they are taking all of your queries on the forum, and they've been preparing very much to have the seamless run of the show. So let me invite the TAs to introduce themselves and also give their brief overview of what kind of research they do. It will also help you understand that the kind of TAs what you have, they're already at a very advanced level of research, and they're already knowing the subject, and therefore they have a lot of interest in conducting this kind of MOOC programs. So I think the next few minutes the TAs are going to share the screen and talk to you further. So let me invite Dr. R.K. Banerjee to share briefly about his research and his TA-ship.
So Arya is joining in a bit. So in meanwhile, myself, Avinash. So I'll introduce myself. So I hope my screen is visible.
Avinash, please introduce yourself and talk.
Yeah. So hello everyone. Myself Avinash Singh. So I'm a fifth year PhD student. So I'm doing this MOOC tail course from the last 2 years, and I have experience of multiple courses like introduction of proteomics and other courses. So meanwhile my screen is coming. So I'll just brief you about. So my work is basically based on the oral cancer. So I work on the oral cancer. So I'm using different samples like saliva, serum, and tissue samples from the oral cancer patients from the Indian population, and then I will performing the proteomics and metabolomics analysis of these samples to try to find the potential biomarker which are involved in the prognosis and diagnosis of the oral cancer. So my major objective is to basically in the oral cancer, there are two major groups based on the nodal infection, so which is nodal samples and non-nodal samples. So I'm trying to find the top classifier which can classify the two group of samples by using a different proteomics and multiomics integration studies, and also validating those classified by using the machine learning approaches and other experimental values. So that's all for my side, and there's a yeah. So Arga, you can introduce yourself. So. Yeah, sure.
So admin, can you allow me to share the screen? Okay, thank you. So, hello everyone, and it has been a long journey for me. I have been PhD as well as the postdoc from the Proteomics lab of IIT Bombay only. I completely means I currently work in the endocrine research in the pituitary tumors, understanding the pathobiology of the various endocrine tumors such as pituitary tumors with the help of the mass spectrometry. And like I am very much means I am very much involved in the clinical research and the PKPD assay. At the end of my PhD tenure, I have couple of publications as well as my patents, which gave me the Dr. Gargi Vishnoi Memorial Best Thesis Award from the Proteomics Lab, IIT Bombay. So here are a few of my papers which have been published in good renowned research articles. And also from time to time from the Proteomics lab, we are involved in the outreach activities, conducting various workshops as well as this kind of NPTEL teaching educational program. So I will be your course TA for introduction to the proteomics. Thank you.
Thank you, Olga. So, good morning, everyone. I am Ayushi Verma. I am also a PhD student in Proteomics lab, and I'm the TA for protein and gel-based proteomics course. And my PhD topic is on comprehensive proteogenomic and metabolic investigation of high-grade gliomas towards identifying novel therapeutic targets. High-grade gliomas are tumor of central nervous system, and glioblastoma, which is the grade 4 tumor, is the most occurring tumor after the meningioma, as you can see here. And this is the most lethal tumor, and we are working on identifying the subtypes within this heterogeneous tumor. And the patient with this tumor has very low survival rate. So we are trying to understand the pathobiology of this tumor, and it is the most malignant tumor of the central nervous system. Towards investigating this tumor, we have performed metabolomics profiling, where we have used spectroscopic based methods, which is ATR, FTR. We have also performed LC-MS based analysis using both the approaches, untargeted and targeted. We were able to identify a few of the metabolites which are mapping to carbohydrate metabolism. You can see acetyl-CoA, sulfosuccinate, glycerol phosphate, and many of the metabolites related to purine, pyrimidine, because the cancer happens due to the metabolic disorders. So various metabolites we have identified in this approach. We have also performed proteomic investigation, where we have performed label-free quantification, and we were able to understand why the high-grade gliomas are so invasive and mesenchymal in nature. And we have identified many of the protein markers like OST, collagens, TGF-beta, which are responsible for the invasive behavior, how the interplay plays within the tumor cells. So that was also our objective. And towards the final objective, we have also performed proteogenomic characterization, where we have performed whole genome sequencing, and it was integrated with the proteomics data, and we were able to identify many of the mutations which were not known even by the TCGA and very specific to Indian population. So we were able to identify certain mutations, and we have actually seen 3 different clusters based on genetic markers and proteomic markers. And these 3 clusters actually have different survival also. So this is a very interesting approach, where we have understood the pathobiology of the disease and also identified different subtypes within a very complex and heterogeneous tumor. And I'm very happy to take this course as a TA, and I'll be interacting more on the forum. So make sure you post your questions on the forum. Thank you.
Yeah, yeah. So good morning, everyone. So thank you, Aishi and others. So I am Ankit. So I will just briefly introduce myself. So I am a integrated MTech PhD fellow at IIT Bombay and currently working under the supervision of Professor Sanjeeva sir in the proteomics lab. And so here I would just give a brief about my research interest and then about the courses of this two courses and uh, how it is helping me to for both interactions and also for the research purpose. So my research purpose, as Ayushi mentioned, that it is to worked on glioma, and meningioma is one of the most occurring brain tumors. So here, what I can say that it is a primary brain tumor which originates in brain and central nervous system and is also can be metastasizing from say. So actually there are basically two types of brain tumor. One is a primary and another is a secondary. So one which is in the originates directly in the brain or CNS, and another is that which metastasizes from other parts. So meningioma is that kind of primary brain tumor with the highest incident occurrence and is one of the growing burdens in the clinical perspective because of its such high incidence rate. So here in my research, what I am trying to find out, so here meningiomas, another major feature is that it remains benign in nature for most of the patients, but certain percentage of patients, it becomes quite aggressive. And for unfortunately, there is no such test available to detect those patients which are going to be aggressive. And for that reason, the management of such patients becomes difficult. So here I have tried to look at the different protein and the metabolite-based markers to identify using these high throughput technologies of proteomics, metabolomics and other omics technologies to identify certain markers which can be used for the detection of those patients that are going to high grades, and we can monitor those easily. Further, we have I understood the therapeutic aspect, because the meningiomas, if you see in the literature that the meningiomas are already treated only with the help of surgery, or it is been radiotherapy, but chemotherapies are uh, not quite effective and only used in the supportive case. So for that reason, our drugs pipeline for the development of the treating of specifically the high grade meningiomas have been developed, and also you can search in brain prot which contains PDTF, so which contains is a comprehensive database of different brain markers, among which in the BDTF there is a comprehensive list of the drugs that are present in different clinical trials of different brain diseases, including meningioma, and further has been tested out in the high grade in vitro through the cell lines. So now coming to the course, so I am the current TA for the course of Introduction to Proteogenomics. So the proteogenomics is a very fascinates me as a research topic, and you can go through these different review articles where they have discussed that how we can approach to identify different new techniques like new antigens, where the major goal remains is to integrate these genomics and proteomics technologies to identify patient-specific mutations and the generation of different customized databases to identify the patient-specific things for the precision medicine. And of course, you are hearing the terms of the precision medicine and precision therapeutics to reduce the side effects and all. So all these can be done with the help of proteogenomics. And as Ayushi was sharing, that how she could identify different specific signatures. So patient-specific signatures, population-specific signatures are all can be used, as well as the recent developments in the mass spectrometry-based uh, not only on the technology, but the software part also is playing a huge role in identification of the proteins, in identification of the peptides. Increasing the coverage is plays a huge role. And I have also been in the past as a have given live session on the Introduction to Proteogenomics as a PMRFTA. You can watch on those YouTube channel. So this time I am interacting as a TA, where I am you can interact with me on the forum or the Telegram group. And of course, and another is the Introduction to Proteomics live session, also I am conducting as a PMRFTA. So you can join live on Friday, 7 p.m. to 9 p.m. for more interactions and discussions. So I will just end by saying happy learning and thank you. Yeah.
All right. Well, thank you, Ankit, and all the TAs, Argya, Avinash, Ayushi, and Ankit. So, students who are watching online, welcome once again for the MOOC live session. Idea was to first introduce you to our TAs who are actively involved in the course, and they're interacting with you on the weekly basis. Today's live session, what we thought, we will make it like a thematic of sample prep and how to perform that in the gel-based proteomics and the mass spectrometry-based proteomics. So sample prep becomes very crucial for any kind of proteomic application. That's the first most important part. And given that one could work on a variety of samples, there is no one protocol which can be best or to be recommended to everybody, right? Even in my lab, we do a lot of clinical proteomics based work. And sometimes, people work on the tissue, looking for the tumor and other kind of diseases. Many times they look for serum or plasma sample, often cerebrospinal fluid, sometimes saliva. So variety of biological samples. And every biological sample brings its own challenge, because the idea is how to best get rid of the abundant proteins, get rid of the nucleic acids, carbohydrates, lipids, salt, and then just try to get the clean protein as much as possible, right? So therefore, to make the trade-off, you have to really understand the biochemistry of your sample very well. And I'm sure, if you are planning to utilize this knowledge in your own research or towards your project, it is a good idea first to know the literature, background literature review about which sample you are going to work on, what is your end goal of proteomics, and then how best you should prepare a sample. So that will be important. In today's session, we will give you, some live instrumentation part from the lab, few small clips. I will also introduce you to save the time. But as the time, if we get more, then we will show you more things from the lab. So let me just, start with the sample prep for the E. coli, and that will help you to understand a basic workflow for the sample preparation.
Bacterial protein extraction. Sonication of the obtained culture, followed by protein extraction. Bacterial culturing. Clean the laminar workspace thoroughly with ethanol and keep the master plate having the bacterial culture ready. Light the spirit lamp or Bunsen burner to maintain sterile and aseptic conditions throughout the process. Carefully remove a bacterial colony from the master plate and inoculate the autoclaved growth media. Incubate this inoculated sample at 37 degrees Celsius for 6 to 8 hours with constant shaking to allow the bacteria to grow. The turbidity of the culture, which gradually increases, indicates bacterial growth. Culture harvesting. Transfer the grown bacterial culture to a fresh tube under sterile conditions. Centrifuge this tube at 12,000 RPM for 10 minutes, maintaining a temperature of 4 degrees Celsius. transfer the pellet obtained containing intact bacterial cells to a fresh microcentrifuge tube. Wash this pellet thoroughly with phosphate buffer to remove any unwanted debris. Sonication. The resuspended pellet is sonicated on ice to enable the bacterial cells to break open so that its contents are released. Sonication involves the use of high-energy sound waves that are capable of breaking open the outer membranes of cells. All cellular contents, including proteins of interest, leak out of this disrupted membrane. Carry out the sonication procedure for 30 seconds with a pulse of 1 second at 40% amplitude. Once it is complete, centrifuge the contents and collect the supernatant that is obtained. Protein extraction. The trisol extraction protocol allows efficient separation of not just the bacterial proteins, but also their DNA and RNA. Add the trisol reagent consisting of quinidinium thiocyanate, phenol, and chloroform to the supernatant obtained after sonication. Mix the contents thoroughly by vortexing. Next, add chloroform to this solution, mix the contents, and place the tube on ice for a few minutes. Centrifuge the tube at 2000 RPM for 5 minutes. 3 distinct layers will be obtained. The topmost is the aqueous layer containing RNA, the center is the interface containing proteins, and the bottom layer is organic and consists of DNA. Discard the transparent top layer having the RNA. Then, add absolute alcohol to the remaining layers and mix the solution well. Centrifuge the contents at 2000 RPM for 5 minutes. The DNA forms a white precipitate at the bottom of the tube, while the proteins remain in the clear supernatant. Collect the supernatant in a fresh tube. Then, add chilled acetone to this tube and mix well by vortexing. Store this solution at -20 degrees Celsius for at least an hour before centrifuging it to obtain the protein pellet. Discard the supernatant and dry the pellet at room temperature. Reconstitute the dried pellet with rehydration buffer and store overnight at -20 degrees Celsius before carrying out protein quantification.
All right, this was one of the type of extraction method. But as I said, depending on the context, one could try different things. Now, let me walk you through about the plasma proteomics. So, many of the biomarkers, for the different diseases, we always look for from the blood, because blood is the most widely tested sample for understanding the human health, right? So if we can identify some disease signature early from the blood plasma, that could be clinically very relevant. But doing proteomics from these samples are very challenging, given that the plasma has a lot of abundant protein like albumin, immunoglobulins, and many other abundant proteins. And they are actually, if you want to do the mass spectrometer-based analysis, they are masking your low abundant proteins, and you will always see in the mass spec the abundant peaks, which comes from these highly abundant proteins. So we try to deplete out these kind of abundant protein, and what you are seeing, like, we are trying to mix them with the column which is having the anti-human albumin or the IgG, so that the abundant proteins can be depleted out. So there are different commercial kits available which can be utilized. But more importantly, nowadays there are more protocols are coming which are trying to remove the abundant proteins without this kind of antibody-based depletion. So again, this is another way of innovation when you can think about sample prep. We can show you a lot, but I think even some students are interested into the plant proteomics. So let me show you this clip of the plant sample preparation.
Select the plant leaf sample of interest and weigh around 300 mg of the leaf on an aluminium foil. Transfer these leaves to a chilled mortar and carefully add liquid nitrogen to it, which helps in drying up the leaves instantaneously. Grind the leaves well using a pestle to obtain a fine powder. To this powder, add around 0.5 ml of lysis buffer containing trichloroacetic acid, acetone, and dithiothiotol, and grind it well until a fine paste is obtained. The lysis buffer causes the plant cells to swell and finally break open, thereby disrupting the membrane and releasing all its intracellular contents. Add another 1 ml of lysis buffer to the paste, and then transfer the solution into a fresh tube after grinding thoroughly to obtain a uniform mixture. Centrifuge the sample at 10,000 rpm for 5 minutes and maintain a temperature of 4 degrees Celsius during the process to ensure that there is no denaturation of the proteins. Discard the supernatant and retain the pellet containing plant proteins along with various other intracellular components. Incubate the pellet at minus 20 degrees Celsius for an hour. Protein precipitation. Remove the pellet from minus 20 degrees Celsius and add chilled acetone to it. Mix the sample well by vortexing to obtain a uniform solution. Centrifuge the contents at 4 degrees Celsius for 5 minutes at a speed of 10,000 rpm. Discard the supernatant and repeat the acetone washing at least 3 times to remove all plant pigments. Obtain protein pellet at room temperature. Reconstitute this pellet with rehydration buffer. Vortex the sample to obtain a uniform solution. Centrifuge the contents the following morning at 10,000 RPM for 5 minutes. Collect the supernatant containing proteins in a fresh tube and store it at minus 20 degrees Celsius until protein quantification is.
All right. So, there could be different ways of extracting the protein. And again, you have seen like how best one could try out different type of methods, especially TC acetone could be one of those methods. Trisol could be other. And we also use quite a bit urea lysis method in our lab. Once you have done the protein extraction, you want to quantify how much protein you are able to get. And there could be again different ways of quantification, but a simple Bradford assay we are going to show in the next.
In order to know the protein concentration in a given sample or to get the total yield of protein in a given sample, it should be quantified. Depending upon the type of lysis buffer used, various quantification method can be used. Today, we will demonstrate you one such method, which is Bradford protein assay. Bradford protein is one of the common methods used for protein quantification, and it is a colorimetric assay. Before performing the assay, firstly, we should have a standard protein. The standard protein here we are using is bovine serum albumin, that is BSA. So here we have prepared serial dilutions of BSA, that is standard 1 to standard 5. And also one is blank, which will involve only the reagent. And here we have one unknown sample. Now, to each of the tube, equal volume of Bradford reagent is added. After addition of reagent, now the tubes should be kept in dark for 5 to 10 minutes. Bradford reagent exists in two different forms. One, when it is unbound, it is in brown color, and then when it binds to protein, it becomes blue color. So the intensity of blue color is directly proportional to the amount of protein present in the given sample. It is basically based on the absorbance shift, which unbound form to bound form changes from 465 nanometer to 595 nanometer. So absorbance should be taken at 595 nanometer. Now, after incubation, each sample is added to the plate. Preferably, the sample should be added in duplicates. Now, all the samples are added in the plate, and then it is ready for taking the readings. For this, we are using multi-scan geo. Plate reader. The first step is to set the plate layout, which includes blanks and all the standards. Also the unknown sample. The next is protocol, in which absorbance at 595 is set. Measurement mode is selected to be precision. And also blank subtraction is set. After getting the absorbance values, the values can be exported. So here we have the absorbance values for all the standards, including standard 1 to 5, as well as the unknown sample 1. So here we have absorbance 1, that is OD1 at 595, and OD2. So firstly, we will calculate the average absorbance. For all the sample, average absorbance is calculated. Similarly, for the unknown sample, average absorbance is calculated. So next step is to plot the standard curve for BSA. For this, sample amount and average OD to be taken. So here we have the plot for standard. Also, the R-square and equation is displayed, and we can see that we have got R-square of 0.99, which means that we can use this equation for the quantification of unknown sample. Using the equation obtained, amount of the protein in the unknown sample can be calculated. So for the unknown sample, we have to calculate X, and the absorbance obtained for the unknown sample would be as Y, as shown in the given formula. Since we have taken 2 microliter of unknown sample, so concentration would be half of it. Now we have got the concentration of our unknown sample. This is how we can get the protein quantified. Its concentration as well as yield can be calculated.
All right. There are a couple of queries on the forum. I just thought to take some of those, and then further I'll continue the session. So Nivit Chandra, welcome. I think you have a lot of interesting questions on the forum. I think you are asking for some more elementary books, which I think TH can help you. They will send you the link. Even I have written a book, Basic Proteomics, based on the MOOC course itself. That could be also helpful to you. Your question is, will not using an IEF much bigger than 24 centimeter provides better resolution? Yes, I think you're right. Like, longer the strip you have, immobilized pH gradient strip, better the resolution you can achieve. And that's where, starting from 7 centimeter, people try to go 11 and then 24. The challenge of going beyond that is there is no commercial strip available, right? If the companies are not making it, you can't use it. If it is available, I think it's advisable to use it, because then you will definitely get bigger separation. You're asking for why does salt causes the bad IEF? You are putting in the isoelectric focusing based on the isoelectric point separation in the electric field. And if the salt is too high, the conductance will raise, and it's going to stop the movement in the electric field. So also, like, the IPG strip what we have, they are having the minute pores, even they get clogged. So bad sample with the salt is always very detrimental for proteomics at different stages, including at IEF. IPG, I think it is the immobilized pH gradient strip. It means on an acrylamide strip, you have different pH buffer, and then depending on your requirements, sometimes you will have 4 to 7 pH strip, sometimes 3 to 10. You can choose those kind of IPG strip, right? Paper wicks are essentially kept on the edges so that the salt during the moment in the electric field, protein moments in the electric field, then the salt can go on the edges and they can be absorbed by the paper wick, which also in between you can change if the IF is going very long, right? You have several small questions. Why not uniform gradient in IEF? You don't want uniform gradient because you initially want a lower voltage, so that the it takes some time for the protein to settle down at the, let's say, 200 250 voltage, and all the salt start coming out. So that's where you start with a low voltage, then slowly you ramp up depending on the strip length, the amount of protein which you have loaded. Considering all of that, it can go to 65,000 volt hour to 100,000 volt hour, and until it completes the entire separation in the electric field. I think that also answers your next question. It is not clear how to align different 2D gels correctly before analysis. That is definitely one of the challenge. Therefore, from the conventional 2D gel, people shifted toward the dye gel, differentiating electrophoresis, where you can have the internal standard. So internal standard will be common across all the different gels, and that could be used for the normalization or the co-detection of the spots. Could a third dimension be utilized? Not much being used in the 2D gel, but definitely looking at the need for separating the proteins in different dimension, different properties. Now, there are different type of mass spectrometry-based platforms are coming, which are also using the ion mobility as another dimension. So now proteomics is coming to 3D and 4D, that kind of stuff to definitely look for much higher separation, right, with different principles. What are gel-free methods? Gel-based, that you want to separate proteins on SDS-based gel or 2D gel or 2D dye gel, or some way of utilizing the gels. Gel-free means you don't want to take the gel and instead you want to separate the complex protein mixture, right? So therefore, you are essentially preparing the sample and then you are injecting directly in the mass spectrometers or other kind of biosensors and trying to do the readout. So that's what it will be, the gel-free, which we are going to show you shortly today. Is it appropriate to quantify based on densitometer? Somewhere I read SDS gels can't be quantified, and how abundant proteins are removed. So I think some of these are very interesting practical questions. Of course, when you're preparing sample, SDS could be the very detrimental. So if the SDS is too high, we try to get rid of SDS with some sort of the cutoff filters. If you have too much urea, we also need to dilute those to avoid again different type of problem with the digestion efficiency, which one would achieve in that way. I think I just see another query from you is about saliency. But before that, there's a how abundant proteins are removed. So abundant proteins are removed if you know which abundant protein you are targeting, like albumin and IgG, and you have antibody against those, which you can do to pull out those abundant protein, and then you can take the entire rest of the protein from the plasma or serum sample. Saliency is actually when we are analyzing the 2D gel, we are trying to define the boundaries of the each spot properly. And how the software will know about it? Software does not know what is the real spot. So you give some parameter like, what is the dimension, what is the diameter, what is the saliency, what is the, you are looking at the intensities of those. And based on that, it puts some sort of, virtual spots on the entire gel. Then you need to further tweak around, and if you make it very small, for example, size and dimension, then it may pick up even the salt and the contaminants. If you make it too large, you are going to miss out the actual proteins. So when you define the proper size and saliency, it is going to look at the kind of 3D shape of the proteins to have the proper boundaries. I hope that's cleared some of the online query. Let me now resume back and continue with the theme which we are talking about, the how to prepare the samples and do the
Other than
proteomics. So I think I covered that. Okay, once you have already done the quantification, you want to run on the SDS page in the first dimension to look at the quality of your protein samples. And this kind of, the SDS page gel pattern, they give you idea, okay, if protein is properly resolved on the across the gel from different samples, or is there any some sort of, contaminants you see, or there is some only abundant proteins you see. So a visual feel of the gels is very important. Then you also do two-dimensional electrophoresis, because if your goal was to do the proper separation on the gel, then I think you can do first dimension separation based on the IEF, and several questions you already asked about it, and then followed by the second dimension, which will be the SDS page. And now you can further separate based on the molecular weight. So once you have done the separation on the gel, or if you want to do gel-free, it means you do not want to separate protein on the gel. You want to proceed further. So if you are using the gel, you will excise the spot or the band from the gel, and then you are going to do further in-gel digestion so that the proteins can be digested to make the peptides. If you were to do the directly from the protein in solution form, you are going to do in solution digestion. So how these things are done, let me just show you in the this short clip.
for the mass spectrometric analysis, in order to identify the proteins in a sample, peptides are required. For getting the peptides, the protein sample should be first digested. Protein digestion can be performed in a gel-free or a gel-based manner. We'll show you how to perform the in-solution digestion, that is gel-free procedure. So now, after quantification, suitable volume of the sample is taken for digestion. Before doing the actual steps in digestion, we need to process through the two steps, that is reduction and alkylation of proteins. For reduction, here we are using TCEP, that is tris-carboxyethyl phosphine. Appropriate volume of TCEP is added. The sample is properly vortexed and spun. After addition of TCEP, that is the reducing agent, it should be incubated at 37 degrees for 1 hour. So after getting incubated at 37 degrees, the samples would be reduced. So after incubation at 37 degrees, reduction is happening. That means all the disulfide bonds are broken within the proteins. So now it's time to alkylate the sample. For alkylation, iodoacetamide is used. During the alkylation procedure, alkylation will block the thiol groups so that the disulfide should not be reformed. After this, the sample is properly vortexed and spun. To perform the efficient alkylation, the sample should be kept in dark for 10 to 15 minutes. So after completing the steps of reduction and alkylation, the next step is addition of trypsin for the digestion. So before that, the step is dilution, which means we first need to dilute the molarity of urea that was present in the initial sample. So the diluent is used for diluting the sample. Appropriate volume of diluent is added to the sample in order to dilute it. Before addition of trypsin, make sure that the pH is 8 so that it can work properly. Now the next step is the addition of trypsin that could be added in the ratio of 1:30 to 1:50, which means that one part of the trypsin can be sufficient for 30 parts of protein. The sample should be vortexed properly and spun. And finally, for optimum protein digestion, the samples need to be incubated at 37 degrees for around 16 to 17 hours. After 16 to 17 hours of incubation, the protein sample is completely digested, and then the sample is dried in order to get the peptide sample. These dried peptides can be taken further for mass spectrometry.
All right. So now you have prepared a sample. Of course, there's some additional step involved as well, which means you have to clean up the peptide before you inject in the mass spec. But now we are taking directly in the lab, and Argya Banerjee will show you how to take these samples and acquire the data using the high resolution mass spectrometer, especially Orbitrap Fusion. So Argya, please display the instrument and discuss about it. All right, so while they're getting ready to show you some actual data acquisition live, idea is like, you take these peptides, and when you are going to put in the mass spectrometer, you have to look for many settings of the chromatography, LC, nano LC, as well as.
Welcome everyone.
All right, continue. Yeah, go ahead.
So welcome everyone. So this is our mass facility at the Proteomics lab, IIT Bombay. I will rather not go to the other instruments at the first lecture. We will go to the one of the latest current age technology, which is a tribrid mass spectrometer. So this is Orbitrap Fusion, in which we generally perform the proteomics experiment. So anything like, as in the mass spec studies, you have seen that there should be a nano LC and there should be a mass spectrometer. So this mass spectrometer is consisting of the three mass analyzer. One is the ion trap, one is the Orbitrap, and one is the quadrupole. And this is the nano LC. So as you have seen, like after dilation, you will reconstitute your peptide samples, and this is how you place your vials. Once your vials is placed in, you will give the algorithm, or you will set the program which is there. And in this like nano LC, you can see all the parameters are easily intact, whether it is a dichloric solvent mixture, the pump A pressure, the pump B pressure. This is your matrix, like where you have taken. Actually, this is A1. That is why you are seeing as the active. You can also easily see like which all methods are there. You can see the graphs, like you can click and you can see the gradient or the Pompey pressure which is coming. You can clearly tabulate all the features of the nano LC which is going on, and you can see like what blank samples, like what samples are currently running. If you want to individually set the programs, you can go and you can set all the programs individually for the nano LC. So once this is done, once you have set the program for the nano LC, it has the tubing. So what this the nano LC does? It does the segmentation, like it has a gradient. On the basis of the gradient, the solvent will be put into the solvent along with the peptide samples will come into the mass spectrometer slowly. So here you can see this is the mass spectrometer. There is a columns. The columns are very important. So I have taken out one column for you. You can see this is C18 column, and this is where from where the spray happens. So you can see the tip of, like, this is the main source of the spray of the columns. So once this is done, this is getting injected into the orbiter mass fusion, where you can see the interference or the front end. The front end which is having the isothermal calibration tube. Once the isothermal calibration tube, the samples are getting injected. Very high voltage are set here, where the samples are bombarded, and you can see the fragmentations which are coming there. So now I would request my colleague Avinash to take over and show how the things are being set up. So this is how the bombardation pattern, and this is a chromatogram. So Avinash, you can tell me.
So after aligning the samples, so we have to look for the parameters, like what parameter is good for the perfect chromatogram. So the first observation is basically we look for the any chromatogram is the Gaussian distribution. So as you can see here, this chromatogram is the Gaussian in pattern. So this is the first observation. So how your sample has been eluted, and you can by this way you can say the sample is good. So now the next step is to look for the MSMS fragmentation. For MSMS fragmentation, you have to just zoom in. And after that, you can see like, so this each peak is representing the precursor. And after that, this small peak is representing your, let me zoom in. Yeah. So this bigger peak is basically the precursor. All the precursor has been acquired at the time at the around the 36.17 tension time. And these are all the MSMS fragmentation. So to further visualize this, you can just click on that and you can see like how many. So if you see the range is around set from the 385 to 1015. And it is these are the total precursors that has been like acquired at the retention time of 36.17. So now if you want to look for the like how many MSMS has been done, so you can see it's all the MS2 fragmentation, and so how many MS1 fragmentation has been occurred. So if you see there's too many MS2 fragmentation is there, that means the data quality is better. And if there is a very less number of MSMS fragmentation, that data is not that great. So these are the two major features we look for any good chromatogram. And now after that, we go for the like static analysis by using and the primary analysis by using a different software. In the next session, the Ankit will brief about those things. So now over to So we can also show you like some of the live run is going for the blank sample. So this is the live run for the blank sample. So this occurs on the basis of the what is the time of gradient. So right for blank we set the 60 minutes of gradient and like time is 44 minutes. So you can see that the upper panel is the chromatogram and the lower fragmentation basically what are the fragmentation happening from the blank sample. So now this is all what we look for in the mass spec, and now over to Ankit.
So before
so we will also give you a brief flavor of how the data analysis is done. So back to you, Professor.
Yes. So before we start on data analysis, just let me take one query from the forum. Om Ingle is asking that he's a student of BTech Food Technology, and how can the knowledge of protein proteomics be useful in the food, right? So of course, these technology, like what you just saw, the mass spectrometer, they have ability to characterize the protein, right? And in the food sector, you have to, at every level, you need to have the good quality check, right? The food should not have any adulterants. It should not have any kind of impurities. It should not get degraded, like the protein content should not get degraded. Lot of shipment issues come, right? The storage issues comes. At every stage, mass spectrometry and various type of analytical techniques are being used actually to characterize those and ensure that the batch-wise food quality is maintained and it is preserved. So I think you will definitely enrich from the experience of learning about proteins and these protein and proteomics-based technologies, right? In a very brief, I hope it gives you excitement that why you are taking this course. So, Ankit, very quickly, please, introduce them to the software. I think we're running short of time. And yeah.
Yes. So, basically, if you have seen in the mass spec that the raw files and the chromatogram, what you see generally, but that you need to understand to identify that what are the particular peptides. So you have seen that you are digesting the peptides, and then you are from the proteins, you are digesting the peptides, and then you are injecting into the mass spectrometer. That is basically you are following a bottom up proteomics. So here is the software. So I think this is one of the software which helps us to first understand from the chromatogram which you have seen to understand and identify what are those peptides. So this is one of the software, that is the FracPipe. So I'm not going into the details, but here after running through these, what you will be getting is that uh, get the matrix of the proteins with its intensity. So I will just show that matrix in a quick moment.
Maybe Ankit, you can move to MetaAnalyst and
Yes,
show.
sir.
Yeah.
Yeah, so this is a kind of matrix which you will be getting, where you have the samples and you have the genes and different kinds of intensities that you will be getting. So now this is the software that is kind of meta-analysis for the secondary analysis. You can use other software as well. This is basically for statistical testing. So what we will do is that we will, according to the matrix that we have designed, either it is in rows or columns, we will choose and we will go to yeah, so you will, wherever your data set is stored, yeah, so you will just upload the file from the path that the data is stored, and you will submit it. So it will, so you can see that after your data file is successfully uploaded, it will contains you the information that you have kept, and you will click on proceed. So there are some of the filterings that you can do for better for your data. So now you will be, according to the particular presence, what are the transformation and normalization you will do, that you will go through the course and we can data. So this is the kind of pattern. So from the Gaussian distribution of what Avinash showed you from the peptides, finally when we identify the proteins and their intensity, this is finally the distribution that we aim for, where the medians align to each other. So now once this data follows this particular pattern, for your simplicity to understand, then we can allow different statistical testing by proceeding. And for instance, I can show you the volcano plot, which shows. So here you can see that there are different kinds of upregulated and downregulated proteins with different intensities, different fold change, as well as the log p value. So you can change it accordingly. You can use FDR, and you can change the fold change threshold, whatever according to your need, and you can see it. Yeah, so this was a quick overview of the data analysis. for the mass spec data?
Of course, this is something which a lot to be done in this area. And in fact, we spend maximum time of analyzing data. Data acquisition is very easy, but what is most important is data analysis and making sense of the data, right? So while I think some of you had already put your questions on the chat, some people have also, posted the questions in the forum and for the form which we had asked them to fill. Let me just take a couple of those questions. What are the evolving challenges for researchers in the field of proteogenomics? So, if you think about, we are looking at proteogenomics analysis of the different clinical samples. So first of all, you need very high quality data, both at genomic level and proteomics level. Then only you can integrate that, right? So while it is very easy to say, but in practical sense, from the same sample, same patient, obtaining very high quality data set is not straightforward, right? At both exome, genome, and looking at the mass spec proteome. So in that way, I think it will be challenging to really acquire for hundreds of patients very high quality uniform data. That is the first beginning challenge itself, right? But then more importantly, you got so much wealth of data. How are you going to integrate that data and then trying to make meaningful sense? That's where your skill of various type of data analysis comes into the play. And more importantly, even biology becomes very important because you need to really think about how to interpret that data. Look for copy number variation, look for mutations, how those mutations are actually important, and also what kind of post-translation modifications you see, what kind of pattern they are making, right? So impact of those mutations at the protein level and the post-transcriptional to post-translational level, all of that need to really comprehend well. So you ideally are looking for a good team of bioinformatician and biologists to make sense of this data. So again, the creating that ecosystem and that skills are very challenging in the proteogenomics scape. Other question is that what skills researcher from biology background they should try to approach to combat the challenges in the field of proteogenomics. First of all, I think good experimental design is important. Thinking about the basic statistics, the power analysis, what will make your study important and the meaningful is crucial. Then you should also think about the skills for high quality data acquisition, and then the different type of programming skills which can make your analysis much more powerful. Open source softwares are pretty good. Many of them are coming forward, but they are still having limitations, right? And how best you can overcome those challenges and do with the programming, that is something which is biologists should try to acquire. So both it's stats skill and the bioinformatics and the programming will be crucial. Another question is about proteins and gel-based proteomics, pre-specified applications. So one of the MOOC courses which we have offered is the proteins and gel-based proteomics. It's a very basic course. In fact, even it's towards your journey of the proteomics and proteogenomics, you should first think about the basic properties of the protein, and then think about separating the proteins in the very simplified matrix of the gels. And whatever you do in the complex proteomics, you always have to use some gels for the separation in the first dimension SDS page. Even you are looking for the protein purity, just you have verified one protein, or the complex protein mixture which you have extracted. Every time we are going to use that for the quality checks. And still one could use 2D or dye gels for various type of quantitative proteomic comparison. So this is a basic course which gives you the flavor of it to get it started, right? I think some more students had some query regarding the how they can make their career in the clinical proteomics area and how they can move from beyond the genomics and transcriptomics field. So of course it's always challenging to look for something very new and acquire the knowledge, but if you're really determined, the proteomics field is very much growing, and if you acquire your knowledge and your training in this area, it is going to be quite rewarding, for sure. Many people, in fact, even sometimes people come for very short exposure in our lab in the proteomics, and they are also very well placed in a lot of industries and different places, given that there is a dearth of these kind of trained individuals. And especially if you can be more comfortable in data analysis, that will make you much more employable. So, think about how best you can, venture into this area. We are always there to help support students, MOOC students who perform very well, who are the toppers of the course. We also offer them opportunities to come to our lab for some short training program or any conference or workshop which we conduct. So you will have multiple ways of interacting with us, right? I think the question about what professional society or organization should I join to stay connected with proteomics. In India, we have Proteomics Society of India, which is a very strong forum for you to join. At the global level, we have Human Proteome Organization, or the HUPO. If you're more keen into the mass spectrometry, then the ASMS is another important forum, right? I think there are many interesting queries, but probably we have to stop the streaming now. And we are, very excited to see like all of you are having so many interesting queries. Just one last query which I'll take. Hariharan is asking, what are the current hot topics in proteomics apart from cancer biology? So proteomics is actually in a very exciting phase right now, given that we have so many advanced mass spectrometers which have come forward, like from Thermo Fisher Astral Orbitrap, and from Bruker there is a timsTOF. With these instruments now, our landscape or ability to look at the proteome has totally changed. We can go very deep now. And therefore, the single cell proteomics has really emerged as one of the important area. Now, from single cell, you can actually extract the protein and do the complex proteome analysis, which is one of the hot areas right now. Additionally, looking at spatial biology, spatial proteomics, how to look at the membrane proteins for the druggable targets, cell surface proteomics. Some of these are the kind of areas which are emerging. Also, like a lot of structural proteomics a field, and also even people are thinking about the imaging-based approaches. Many of these are, quite exciting, which is the new hot topic and development in the field. All right. Well, thanks everybody for staying connected, and some very nice and interesting questions on the forum and in the form. We will again interact with you in the next live session, which will be in September. We will announce you further. In fact, I think I have a date already. So let me share my slide and show you. So it is 15 September, which we will talk more about multiomics and big data analysis. I'm sure you'll have more excitement and more exciting things to ask us, and we will show you again live from the lab some of these experiments. Thank you very much, and I hope MOOC course is giving you a good start, and will be there to make it more and more interesting for you. Thank you very much, everybody, and all the TAs who have been part of today's live session.